So in the very last part of Chapter 4, we're going to go inside the bacterial cell. So again, we're working our way out in. So we've talked about appendages, we've talked about the cell envelope, and so now we're going to look at the things that are on the inside of a bacterial or prokaryotic cell. So first we'll notice the cytoplasm. Now we know all cells have this. This is that semi-fluid that's found in the cell. It contains sugars, it contains amino acids, salts. It's also high in water. And so the overall purpose of this is it basically serves as a location for all the contents of a cell. Also helps to kind of maintain the shape of the cell as well, and it provides some important materials that are needed in metabolic reactions. So we also note that, remember, prokaryotes do not have a nucleus. Instead, they have what we call the nucleoid region, and in that region or area you would find chromosomes and plasmids. Now, chromosomes are still segments of DNA, just like they are in our chromosomes. The difference is their structure. So for us, we know that our DNA, it's a double helix. We know that our chromosomes are very linear. But when we look at bacterial chromosomes, they're single, so they're much smaller. And instead of being linear, they are circular. Okay? They still contain DNA, just like our chromosomes do. We also note plasmids in this area. Now, plasmids are interesting because the definition of a plasmid is it's a non-essential piece of DNA. Now, non-essential just simply means they don't need it for growth. They don't need it for metabolism. It's essentially extra DNA. Now, why in the world will we make a point to mention just something that's non-essential? Well, this non-essential piece of DNA can actually be transmitted from one bacterial cell to another. And so this happens a lot in conjugation, where they're swapping genetic material. Sometimes they'll swap these plasmids. And the benefit to the bacterial cell, not necessarily to us, but to them, is that these plasmids can carry manipulated genetic information. Now, again, why would that be important? Well, that manipulated genetic information could be drug resistance. And so these plasmids allow cells to take their own drug resistance and give it to another cell. Now, typically, drug resistance is based on exposure. But because they can pass the drug resistance from one cell to another, you could essentially develop drug resistance when you've never actually been exposed to that drug. And so this is presenting a problem for us, which is one of the reasons why we're seeing more drug resistance now than we've ever seen before. Also inside of a prokaryotic cell we'll find ribosomes. Now we learn in biology that ribosomes are the location for protein synthesis. Same function here. The difference has to do with size. So we'll note that in bacterial cells they are much smaller than they are in eukaryotic cells. But they're again found all inside of the prokaryote or the bacteria cell. And again, same function of making proteins. Also inside of a bacterial cell you'll find what they call inclusion bodies. Inclusion bodies are basically vacuoles. They're storage vessels that are found inside of the bacterial cell. They can store nutrients, they can store waste, they can even store gas. And so for some prokaryotes the gas inside of them actually helps them float, increases their buoyancy. So a very important part of a bacterial cell. Also, inside of a bacterial cell you can find a cytoskeleton. We talk in biology, the cytoskeleton for a cell basically does the same thing that your bony skeleton does for your body. It provides structure, it provides support, and it also helps to encourage movement. So that does the same thing here for a bacterial cell. Also, we'll note that some cells have the ability to form endospores. So a couple things we'll note about endospores. First off, endospores are not developed by all cells. It's only a certain group of them. So those found in groupings of Clostridium, Bacillus bacteria, and Spore Sarcinas. Endospores have a two-phase life cycle, or bacteria that form them have a two-phase life cycle. So they have what we call the vegetative cell, which is the metabolically active, the growing, the reproducing cell. And then the endospore is what we consider to be the dormant, non-active phase, where they're able to survive unfavorable conditions. You know, think about bacteria, they love warm, they love moist, they love dark. And so as long as they're in those conditions, they're vegetative, they're metabolically active. But if they get in an environment that's not favorable for growth, they get in a cold, dry, bright environment, a lot of bacteria are going to die unless they can form an endospore, which basically is a dormant shell that allows them to survive until conditions become favorable again. The two vocabulary words they mention here: sporulation is simply when they form the endospore. Germination is when they leave that endospore and return to the vegetative growth. And so just two terms that they mention there. Now, why are endospores important? Again, it allows them to become metabolically inactive and to resist unfavorable conditions. Now, here's the scary thing about this. Endospores can actually resist boiling. So if you boil something, that doesn't mean it's sterile. If you had bacteria on it that could form an endospore, they can actually resist the temperatures of boiling and maintain survival until conditions become favorable again. The only way to get rid of an endospore is to put them in something like an autoclave or a pressure cooker. Pressurized steam that gets up to 120 degrees Celsius with 15 psi, so pressure, for 20 to 30 minutes will destroy them completely. So again, endospores not used for reproduction, but used for survival. Last thing we'll mention in this section has to do with shapes and arrangements. So we will mention bacterial shapes and sizes. Typically they have three basic shapes. So we see coccus, bacillus, and spirilla. So coccus, we know this is the spherical form. Bacillus is rod-shaped, and then spirillum is corkscrew or twisted. So if you've heard of Staphylococcus bacteria, it's shaped like circles. Bacillus, like E. coli, is shaped like a rod. And then you probably have not heard of a lot of spirilla bacteria, but we'll look at Acrospirillum and things like that this semester. So you can see there's your coccus, circular shaped. There's your rod or bacillus shape. And you can see there's your spirillum, which is kind of corkscrew. So we also see, and they mention pleomorphism, the ability to change shape. What we're going to be dealing with this semester, though, will be bacteria that don't change shape. So we'll deal with those that have a basic shape with them. We also know that bacteria typically arrange themselves in certain patterns of arrangement. So they can be found by themselves. Diplo means they can be found in pairs. Tetrad arrangements are groups of 4. Or then you can get what we call clusters or chains. Now, some vocabulary words that we'll look at in lab, the vocabulary word for cluster is typically staph. The vocabulary word for chain is typically strep. So if you look at Streptococcus bacteria, it's chains of circles. Or Staphylococcus, it's clusters of circles. And the bacillus can take on some of those same patterns of arrangement. Now, one last thing that it mentions in Chapter 4 are comparing domain archaea with domain bacteria and eukarya. We mentioned this earlier, so I would refer you back to Chapter 1. If you have further questions about this objective, though, you're welcome to ask in class.